All right. Um, as part of this class, like we're doing biology while kind of learning how to use this tool, right? And so I think I have this class partially designed to basically learn by doing. All right, so not all of these tricks, not everything that we're talking about today, or in this class period, will translate into your careers or anything like that. But the point is that we've looked in books and we've heard these concepts described, protein coding genes or microRNAs or anything like that. But now with the computer, we have the ability to go find that stuff and go look at it and examine behaviors of genomes and other things. So that's kind of what we're really just going to spend the last couple of days of this first half of the semester on. It's just kind of playing around with it, with the human genome, and seeing what we can see, and using the tools that we've learned up to this point to do so, but also introduce some other tools that help us play around. So if y'all are in this class, I have a file that I made earlier today. It's in data, and so it's this hg38.tes.tab. And so that's a file. It has all the coordinates of transposable elements in the human genome. So if I head this real quick, right, it just tells me this is itself would be called a bed file. And a bed file really is just three things. Three things are important in it. There's the chromosome, the start position, and the stop position. And those are the only three things that matter. But beyond those three columns, we can add optional columns, right? Like orientation of the gene or annotation. Is it in the 5 to 3 or 3 to 5 orientation given the reference? And also what's present. So satellites, lines, DNA signs, etc. Right? It's just a bed file. And GTFs are similar to a bed file because the first three columns will be the same, just the chromosome number, the start position, and the stop position. Right? Always going to be the same in a bed, GTF, GFF, or usually a VCF as well. Always relevant. But no, so you can copy this over to your home and play around with it and follow along with what I'm going to be doing. Again, just learning by doing. Like what's in the genome, learn by doing. So I'm going to go back home and copy that over. So I'm living in CD test GTF. That's where I'm living. And so I'm just going to copy that over, so that I have it as well. And so it's kind of large. I've reduced it down a little bit. 156 megs, not bad. Not the largest genome file that we've made. And another difference that might be another difference between where I stopped and where I stopped in the last video and now is I unzipped this HG38FA. Oh no, that's not it. I unzipped the GTF. So I just gun zipped the Homo sapiens GTF. Where am I at? It won't let me because that's not a gzip file. Anyways, so those are the only things that are different. I still have my human gene GTF file that's just all the locations of the start and stop of every single protein coding gene. And now I have a TE file as well. All right, so let's see what we can do, what we can pull out of some of this information, out of these massive files that are impossibly large to actually look at. And so I can introduce another special character, I should say. Is the up caret, and so this will be a Shift 6 button. And so what this character stands for is start of line. So if I ask for MT, so for example, I grep for MT in human gene GTF, right? It's just going to pull out all lines that have GT in it or MT in it. So we can even kind of do that, right? So nothing irrelevant. It's just going to give me any line in this file that has MT in it. That's curious. MTHFR. I wonder who named that gene. That's an interesting one. Yeah, anytime, just MT just gets spit out. Any line with MT in it. But if I put the up caret in front of the MT, now I'm asking to grep out any line that starts with MT. Right, so now as opposed to just lines that have MT in it, now I'm just going to grep out lines that start with MT. And so that's going to be substantially different. Pop out all the mitochondrial genes, because these are the only lines that start with MT. And so this can be true for any chromosome that we care about, right? So that's what's nice about this, is that we can decidedly grep out chromosome-specific annotations. Right? I tried to grep out mitochondrial genes last time, and I forgot about the up arrow or the up caret character. So now, if I actually want to accurately count the number of genes, protein coding genes on the mitochondria, grep MT on that, and then WC. And now it's going to tell me there are 13 lines in that, so 13 protein coding genes on the mitochondria. Right, and there they all are. Right, so you'll see this ND pretty common. I forget exactly all of the and for, but they're all going to be genes related to cell respiration. Right, all mitochondrial genes are going to function in cell respiration. They're either going to be in the electron transport chain or Krebs or something like that. But that's site B, ND4, yeah, CO3. I think that's oh, I don't want to say anything. I'm going to embarrass myself. ATP synthesis genes, right? But yeah. Anyways, we can do the same thing with any chromosome that we want. One will be a little tricky because we have chromosome number 1, but we also have chromosome 10, 11, 12, 13, etc. So that's not going to be very helpful if I want to grep out chromosome number 1. But we can certainly grep out all genes on the X chromosome. And we should see that there's going to be a substantial difference in the number of protein coding genes on the X chromosome 800 and the Y chromosome 46. Right? So this stuff can be found in textbooks, but we can just go find it ourselves using this. And so 46 isn't a huge number. So let's see if we can go through that. And I'm curious if this will be, yeah, SRY. And so I talk about the SRY gene in like intro biology classes, and I think also in genetics. But right, this SRY is the maleness gene. It basically causes real fetal development, right? So in fetal development, it causes maleness in fetal development. So embryos with this SRY gene expressed will develop male, right? It's called a maleness gene. But we know the exact location of it. It's on the Y chromosome at that base pair position. So that would be 2.7 million bases in. We'll find the SRY gene. Right, any single protein coding gene on the X chromosome or on the Y chromosome is here. Right, everything gets annotated. And so like anything that we want to look for, we can find. Maybe one more. We'll grep out microRNAs. So as I mentioned, I have two GTFs. I have just the protein coding genes, just the stuff that I've grepped out with the protein coding genes, but I also have the raw GTF downloaded from Ensemble. So grepping on a word isn't very successful. So if I just grep out microRNA out of the raw GTF, I'm going to pipe it to less so I can scroll up and down through it. But it tells me, it gives me a lot of transcript information as well. So it gives me the gene location, but it also gives me a transcript location. And it gives me like transcript information, it gives me exon information, a lot of stuff that I don't actually need. I just want to know where that microRNA is at. And it seems pretty successful to just grep out any line that has transcript in it. That seems pretty successful. So now we can actually look at the microRNAs that are present in the entire human genome. So we might as well just count that real quick with our WC. And so about 1,800 microRNAs are found in the human genome, and that's pretty accurate. I've seen estimates that that's a little closer to 2,000, but that's close enough. 1,800 and 2,000 are close enough for me. So it's accurate enough, but we might as well look at that as well. All right, so this MERBase refers to a database. So you can get this microRNA. You can get this microRNA out of MERBase. MERBase is just a database that holds on to microRNA sequences and information. But this tells us where exactly all of the microRNAs are found in the genome. So all of these are on chromosome number 1. Doesn't look like we have, at least they're not annotated, but it stops at 22. So the last line of this file is really even 21. So it seems like, based on this information, we don't have any microRNAs on chromosome 22, the X or the Y. They're just going to be between 1 and 21. And so if we remind ourselves, microRNAs, they're fairly diverse. And what we know is when they're transcribed and processed and functioning, they're going to guide like agoproteins or some kind of a protein to the 3' tail or the 3' UTR of a transcript, and ultimately prevent translation through some mechanism we don't fully know. I don't think we fully know yet. But microRNAs will form a complex with an ago, bind to the 3' UTR and prevent translation, either through like cleavage or just interrupting a ribosome, things like that. So we can find all the microRNAs that we ever want to. And again, the human genome has an advantage because it is so well annotated, right? If we look at some of the non-model stuff, you're not going to find this level of annotation or this level of detail in our GTFs. Like, we don't know all the microRNAs in every single species, right? But certainly we know them in humans. All right. Yeah, so maybe taking a step forward. Let's see. I guess one last thing to look at are pseudogenes. I think pseudogenes are awesome, or at least some kind of pseudogenes, specifically processed pseudogenes. So if we look at this, let me just grep out pseudo real quick, because we can only grep out words that are present in a file, right? We can't grep out anything that we want. We can only grep out words that are present. And so I know pseudogene is in here. I know pseudo is going to be somewhere in here. So I'm just going to grep pseudo real quick and see variant. So all we see here, so we do see transcribed unprocessed pseudogene. transcribed unprocessed pseudogene. But recognize that at least pseudogene has an underscore in it. And so there is a difference between unprocessed and processed pseudogenes. But that's kind of partly for y'all to figure out what that means. But if I want to find, say, specifically processed pseudogenes, I at least have to make sure that process and pseudo are joined by a underscore, right? Because that's how it appears in these files. And so let me just see what happens there. It's the same thing. Let me less for it. Okay, so yeah, this isn't transcribed. It's just an unprocessed pseudogene. And if we see a pattern here, there we go. We both, if we grep on process, we get both unprocessed and processed. So if we want to get rid of that unprocessed, I might grep V on the un. Let's see if that gets rid of it. Yeah. So now we're just left with the processed pseudogenes. Yeah, processed pseudogene, etc. And so processed pseudogenes are kind of awesome because they're kind of a byproduct of transposable elements. And processed pseudogenes are why there's like this whole discussion of like mRNA vaccines integrating into our genome. But that's all of that's kind of related to transposable elements, and it's kind of a neat thing. I think they're awesome. Just seeing their presence is weird. Anyways. Recognize I'm not saving any of the output. I'm just kind of exploring around, but we certainly can. Like for example, I certainly can save all of the location of microRNA. So grep on the mRNA, grep out transcript, and save it. Say human dot miRNA. There's definitely a difference between mRNA and microRNA. .gtf. Yeah, there they all are. And so, like, just from this one file and just understanding one command, we can find all of and have ready for processing all of the microRNA annotations, at least present in Ensemble. Fancy. What else can we learn about these? And so, if you know anything about me, certainly now, you know I like TEs. Like, transposable elements are so weird, and they make up such a big chunk of the genome. We want to know what's going on with them. We want to understand better why they're there, why they go away, and what kind of impact they have on genome integrity. Even their effects on speciation and things like that. They're interesting. Anyways, so one thing we can do, this is kind of interesting, is like I don't think in undergrad biology we really don't take advantage or appreciate transposable elements. They definitely get overlooked pretty frequently. But we're going to use this information from two very different files. So we have the HG, so this is the TE file, and our GTF. So I'm just gonna, for now, for like 2 seconds, I'm just gonna play with the protein coding, the genic information, so the human gene, GTF. Okay, so we have the two different files, but the nice thing about those GTFs is that we can find overlapping regions, right? So it's kind of tricky. So if I want to say, okay, I have this whole genic region of OR4F5. I have this whole coding gene that's about 6,000 bases long. I can ask, are there any TEs in this locus? Right, and I can do that with this tool called bedtools. Bedtools. And so bedtools is this, might be one of the most convenient tools invented in bioinformatics, and quite honestly, it does some of the most intuitive stuff. And the authors who wrote it, I think it was a grad student, maybe a postdoc, but I want to lean towards a grad student. Wrote bedtools, and it's become one of the most commonly used tools in bioinformatics, because the steps of finding like overlapping regions or annotations that overlap is hard to code out on your own. It's a little tricky. It seems intuitive, but it's a little hard to do it on your own. And even the author was hesitant about releasing it. He wrote it out for himself in his lab, and then only kind of later, like I think he made it available before he published an article on it. And I think he wrote a summary on the article, and it's one of the more cited articles in bioinformatics. I'm just curious. Yeah, bedtools. There's bedtools in Google Scholar, and so bedtools is up to about 12,000 citations. So at least 12,000 different people have used it, but way more than that. It just appears in 12,000 different articles, and it's only 10 years old or 11 years old. That's a very, very, very fast rate. But yeah. Well, he's a professor now. He's at Utah. But right, it's the most cited paper, by far. Most cited paper is bedtools. Anyways, I digress. So using this tool, like it can do so much. It really can do so much. But what I'm going to use it most for is this intersect tool. And even further, I don't have to write out. So how this is kind of written, it's like you call bedtools and then the subcommand. So as opposed to like calling an option, like -A or -V or anything like this, if you were to call it, it'd be like bedtools intersect, right? Get that up. And that works fine, right? It's gonna, now we have all the options of how we can use bedtools intersect. But there's a more convenient way to write this out, is to just write out intersect bed. And you should all have that. That should be accessible to anybody using the server. Same thing with intersect bed. Yeah, so all of that's available to y'all. Okay, so again, the nice thing about this intersect is we can read one file, read another file, and find where annotations overlap and print that stuff out. So for example, whenever I'm doing this intersect bed, we have to call the two files, and so we call two files with -A and -B. This is in the manual, but I'm just going to kind of just briefly go over it. And so whatever file you list for B, that file is going to be read into memory. Now we really haven't talked about like all the parts of a computer. But right, you have a hard drive, you have your memory, a motherboard, and your processors. There's also a graphical processor. I mean, but that's generally about it. Your hard drive and your memory. And so whenever you're actually interacting with any kind of file, say you're opening up a Word doc, what you're basically doing is moving that file or creating a copy from your hard drive and putting it in your memory. And so now you're using that memory stick that's like 4 or 8 gigabytes, give or take, whatever's on your computer, to look at that thing. And so memory is much more limited than hard drive space. And so whatever file is in B is what's going to be opened and stored in memory and kind of saved there. And then the other file is going to be scanned and compared to what file is stored in memory. And so when using this tool, when using bedtools, the smallest file should be B. That's what should be listed for your B option. So if I'm looking at these files, so I have my TE file and my Gene GTF. So I'm going to use my Gene GTF because this is much smaller. That's going to be my B option. So intersect bed -A will be my HG TEs, and -B will be my human gene. And so by default, what's going to happen is like, what will happen by default? Yeah, it's just going to print out all the contents of A that overlap with B. Right, that's the default behavior of this. I'm going to less it so it doesn't spit out. So this is all the TEs that overlap with coding regions, or at least protein coding genes, right? But say I want to see the exact location where what overlaps with what. With bedtools, I can do with WA and WB. These two additional options are basically going to say write WA and write WB. Where file A overlaps with file B. Just go ahead and write that out. And so I'm just going to less that. And so half of this will be dedicated to the TE file, and the rest of it is the coding gene file. Right? And so what we're kind of seeing is a lot of TEs overlap with protein coding genes. Weird. However, recognize all that I'm looking at here, the start of transcription and the end of transcription. But remember, everything gets transcribed. The UTRs get transcribed and the introns get transcribed. And so most likely, few protein coding exons that are going to overlap with TEs are in almost every single protein coding gene, UTRs or the introns. Almost every single protein coding gene has a transposable element within its border. Usually not the exons because that's very disruptive, but certainly in the introns. And honestly, sometimes, on occasion, you can find a T. It can happen. And so that's kind of handy. And we can go further. If I wanted to look at the Homo sapiens, like the full-on GTF, I'm gonna change my WAs and WBs so that my TE is what's read into memory because it's the smaller file. So if I'm gonna read the whole thing, HG38 TEs, and the raw GTF, since the raw GTF is considerably bigger, 1.3 gigabytes, I don't want to load 1.3 gigabytes into memory, right? I have that much memory to do this, but I really don't want to. And certainly on a small computer, we really don't want to do that at all. It's an easy way to break your computer is to overload your memory. And this will probably take a couple of seconds to actually go because we have 1.3 gigabytes of data to process. And again, if I were to try to do this by hand or even write out my own script to find overlapping regions, I've tried it. It takes so long. It takes so long to process anything. I'm not good enough with computing to do that. And that's the joy of bedtools, is it lets me and other nerds just learn how to use Linux and learn how to use the tool and not have to worry about coding. And that's always an advantage, is when you don't have to code and process data. That's always an advantage. But yeah, let's see. So we have a few, right? We have a few TEs and unprocessed pseudogenes. So that's true. That's a whole unprocessed pseudogene, and it's going to overlap with a line. Right, so let's see. So this runs for about 15,000 bases. And for a really small fragment, that's a small TE, right? That's a very small fragment. That's only like 100 bases of this line element. So that barely even counts. But nonetheless. Let's see see link RNAs. So this is kind of neat. Long non-coding RNAs. And so we will see like it will be given an exon. So let's see if I can even find out exon. So while I just so you know, while I am in less, I can run a search function. So I can do I can run a find command. And so I can start a find command by using I think my backslash. So the button with my question mark, and then the word I want to find. For example, backslash exon will find any time that exon is in place in this file. But even though I have exons here, they're all related to long non-coding RNAs. And so what's kind of neat about these long non-coding RNAs is that they are pretty long, right? It can run several thousands of base pairs, even like 50,000+ base pairs. They can be crazy long, and they're full of TEs. Long non-coding stuff are full of transposable element fragments, and I don't think we're entirely sure. It's certainly not every long non-coding RNA region. We know what some of them do, but certainly not all of them. But nonetheless, they're full of TEs, and there's a huge bias for TEs and long non-coding stuff. Right? Almost any time I look for exon, it's going to be linked to a long non-coding stuff. So I doubt I could certainly get this more complex and try to just grep out exons and overlap them with TEs and see what happens, but I don't think we need to do that today. However, the last thing that I'll do with intersect bed, this bedtools tool, is we can also kind of use it like grep. And so we can print the stuff that overlaps out, but we can also print the stuff that doesn't overlap out. And so let's see, if we look at the help menu, let's see that option. The option is -v. So very much like grep -v, right? To print stuff that doesn't have that line in it or doesn't overlap. And so what it's going to do is report entries in A that doesn't overlap B. So for example, let's say I want to find all the genes that don't overlap with TEs. So this is where I will change. So I'm gonna use my intersect command. And so A, what I want to print out of A, so my A is going to be my gene file, human genes, and my B will be the TEs. And again, right, I'm forced to put the larger file in B because I want to see stuff that doesn't overlap with A. And so that will just print out the things that do overlap. It's going to take forever. But if I use my -v option, now it's going to print out stuff that doesn't overlap with a TE. It's going to print out genes that don't overlap with a TE of any kind whatsoever. Let's see how long that takes. Not that long. But here we go. We're going to look at this for. We're going to count how many genes. WC. Going to count how many genes don't overlap with TEs. $1,300. That's it. Just 1,300 genes. Remember, there are about 20,000 of them. 1,300 don't have a TE anywhere near them. Not in a UTR, not in an intron, nothing. So that kind of tells you something, that TEs are everywhere. They're in almost every single protein coding gene. Again, most of the time they're in introns, but they're certainly there. And let's just do this one more time. Let's just find all the TEs. Let's just save a file that has all the TEs that don't overlap with a gene, right? Let's just say they don't overlap with anything in the GTF file, in our raw GTF file. So again, we're going to use the -v option. And so I want to print the TEs. I want to print the TE, so my HG38. And my B, again, I'm forced to do this. At least I have enough space to do this. I have enough memory to do this. But again, I'm forced to do it, not because I want to. It's just I want to see the TEs that don't overlap anything in the human GT and among human annotations. And so that will take a while. So I'm just going to save this as like I'll just say non-anno TEs. And so this will still be I'll call it a tab file. And so that will take a couple of seconds. And what's going to take so long is just uploading that giant GTF file into memory. That will take longer than actually processing this, but loading that, moving it from the hard drive into memory will take the longest. And that's why I don't put the largest file as B, just because it takes longer. But let's see. Okay. Now let's just compare the sizes of these two files. So here's our raw TE file, and here's our file that doesn't overlap anything. And so it's less than half, right? Less than half of TEs don't overlap something. And if I cared, then I might play with those. I might look through them. I might play with them. I might see if there's a bias for a line or a sign or something like that. There's probably not. There's probably no real information in it. But at this point, I certainly could check. And you know what? I might as well. This isn't a massive file, so let's. I'm gonna do. I don't think I'm gonna require y'all, and I'll explore these commands later, probably on Friday. But cut lets me cut columns out, and so I can define a column number. Let's see, non-anno. So let's just say I want to count the number of times each of these annotation pops up. So each time line L1 comes out, I want to count how many times that happens. So I'm going to cut out this 1, 2, 3, 4, fifth column. So cut-F5 is going to print just the fifth column of what I call this, non-anno. Right, so it just cuts this out. And then I'm going to pipe it to another command that's sorting. So what it's going to do is it's going to order everything out. So everything that's a line -L1 will be printed together. And everything that's assigned MER will be printed together, etc. And lastly, I'm going to do a unique -C. So instead of printing out sort, all the products of sort, and there might be 10,000 lines in here, I can just print out one unique line, and then tell me how many times that was present with this unique -C. So I'm just going to let this run. It'll take a while. The sorting algorithm will take longest out of anything here. But yeah, here's our output. And so let's see, this DNA hat was present 25,000 times. This DNA Charlie was present almost 100,000. This line, a line L2, so there we go, line L1 is present 450,000 times in unannotated regions. Right, you don't even have to be good at coding to do genomics. If you just get clever with the tools at hand that Linux provides, you can learn about the contents of a genome. There's a sign, there's two signs, these are the most abundant signs. This ALU sign is mostly primate specific, and it's one of the most common signs in the human genome anyways. But yeah, it was present about 500,000 times as well. So definitely, of all of these things, I could take this another step further and sort based off of this number and find the most common stuff, but we won't worry about that. But it seems like just a quick scan, the most common TEs, the line 1 and the ALU were the most common. Where's that ALU at? Yeah. And so that just so happens that reflects the background of the genome anyways. ALUs are super abundant, and so are L1s. They're super abundant, right? So nothing really biased. It's probably just going to reflect the background of the genome anyways. But nonetheless, I'll probably play around with this cut, sort, and unique a little bit more. But yeah, these three commands piped together. If I'm on the computer, I'll do this almost at least 5 times a session. Really common for me exploring files is to use cut, sort, and unique. Anyways, so I'll call it there. We'll do this again on Friday. We'll play around with GTFs, play around with these commands, see what we can find out. And it's spring break, so we won't have to do anything next week. All right, so I'm going to kill this now, and I'll see you all again on Friday.